Hello, everybody. We would like to thank you very much for your interest in our project entitled Segregating the Prefrontal Cortex by means of deep brain stimulation. We are Barbara Hollunder and Ningfen Li from the Network Stimulation Lab at Charité University Medicine Berlin, which is headed by Andreas Horn. In research into deep brain stimulation, or DBS in short, it remains a conundrum why stimulation to the same and relatively small subcortical target, namely the subthalamic nucleus, can be an efficient treatment for different disorders of quite heterogeneous symptomatology, ranging from Parkinson's disease to dystonia, Tourette syndrome, and even obsessive-compulsive disorder. One key to resolve this question may come from the fact that the subthalamic nucleus receives widespread external input from the entire prefrontal cortex. Despite a considerable amount of cross-communication, these circuits and even focal projection sites within the nucleus itself can be clustered to some degree by their relative preference to process sensory motor, limbic versus associative information. Intriguingly, most of these insights actually stem from lesion and DBS investigations, which have helped to map network-level effects of neuromodulation across different disorders on the group level. Hence, within the scope of the study, we intended to leverage optimal connectivity profiles of DBS electrodes to the subthalamic nucleus in 4 different disorders as a means of systematically mapping the functional organization of the subthalamic nucleus and prefrontal cortex. For this purpose, we were able to pool data from 8 patient cohorts across international DBS centers, which each received bilateral DBS to the subthalamic nucleus for treatment of either of these disorders. We employed their respective main assessments to measure clinical improvement. Using lead DBS software, we first reconstructed DBS electrodes and estimated stimulation volumes. Then, 3 conversion methods were performed, which included DBS sweet spot mapping to look into focal stimulation sites associated with disease-specific clinical improvement, but also DBS fiber filtering and network mapping to investigate network-level effects at both a voxel and on a tract level. Indeed, not only stimulation sweet spots within the subthalamic nucleus itself, but also beneficial fibers and cortical projection sites followed a comparable pattern of functional organization. So, while a certain degree of segregation appears to remain intact, our results may also be reconciled with the idea of spatial information compression at each spatial level of the brain. This again could be the reason why we can modulate large-scale brain networks to treat heterogeneous disorders via single electrodes if we place those at integrator hubs such as the subthalamic nucleus. To summarize, we hope that we could convince you of the potentials of connectomic DBS, which may indeed be used as a causal flashlight into the functional organization of the human connectome at all levels, between cortex and subcortex. Finally, we would like to thank you and the organizers very much for your interest in our project, as well as our collaborators for generously contributing data and expertise.